The FHAB technology significantly extends the half-life and targets tumor and lymphatic tissues, providing a mechanism for dose sparing and an opportunity to improve the safety and efficacy profile.
Welcome back for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR. Today we are excited to welcome Sonnet BioTherapeutics to our Virtual Investor platform, and I am very pleased to be joined by Dr. Pankaj Mohan. He is Chief Executive Officer of Sonnet BioTherapeutics. Welcome, Pankaj.
Thank you so much, Janine. I'm happy to be here.
We're always happy to have you on our platform, Pankaj. Before we get started, I just want to inform our audience that Sonnet BioTherapeutics Holdings, Inc. is listed on NASDAQ and trades under the ticker SONN. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's website at sonnetbio.com or the SEC's website for their latest filings and information. So, Pankaj, we're going to dive right in. Let's talk about some of the attributes that have been astutely designed with SON 1010, starting with the prolonged half-life of the molecule. And why is this important compared to the wild-type IL-12?
Thank you, Janine. We are pleased to share these findings, which is the first time we have fully described the extensive research that was required to discover and develop our signature FHAB platform, or fully human albumin-binding platform. And importantly, this has been peer-reviewed and published by a leading journal of Frontiers in Immunology. I will just give you a brief on the discovery effort that happened years ago. The discovery program was designed to screen for single chain variable fragments called SCFV, domain that strongly bound albumin from mice, humans, and a certain type of monkey, as this is the key for tumor targeting and retention. These were developed through a series of steps to identify strongly binding molecules that bind tightly over a 5.8 to 7.2 pH range inside the tumor, which is the tumor environment, and don't interfere with the normal physiology of albumin to bind the neonatal Fc receptor called FcRn. This result is having a prolonged half-life in serum and binding to SPARC and GP60. These proteins allow albumin to target the tumor microenvironment, or TME. From all this research with multiple of these molecules, one molecule was selected from several initial candidates, and a single mutation was introduced that minimizes the potential for immunogenicity. This FHAB domain was characterized and manufacturing processes were developed to prepare Sonnet's first drug candidate for the clinic. The composition of matter claims on this FHAB domain and SON 1010 fusion protein have been issued in a number of major markets, including but not limited to the US, China, Japan, Russia, and New Zealand, and expires between 2038 and 2039, giving us a long window for us to can develop this forward. This work also lays the foundation for each of the products on our platform that are designed to safely extend the half-life of cytokines and deliver them to the tumor, where they can convert the immunological response from cold to hot, demonstrated in preclinical models, where tumor shrinkage was improved over 35 times as compared to an equivalent dose of wild-type interleukin-12 at the day 10 in a mouse B16F10 melanoma model. Hence, what you see is that compared with the wild type, it tremendously improves the performance of the molecule with a cancer tumor by by shrinking it 35 times faster and shorter as compared to the wild type interleukin 12.
Extremely helpful, Pankaj. So the next attribute discussed in the paper is the targeting aspect of SON1010. Can you help investors understand how SON10 specifically targets the tumor versus healthy cells and what this means for patients?
SON1010 is a candidate immunotherapeutic recombinant drug that links unmodified single chain human interleukin 12 with a single chain antibody fragment called A10M3, our fully human albumin binding domain that we call the human FHAB or fully human albumin binding domain. This was selected in part because it binds albumin both at normal pH as well as at the acidic pH typically found in the TME or tumor microenvironment. The FHAB technology significantly extends the half-life and targets tumor and lymphatic tissues, providing a mechanism for dose sparing and an opportunity to improve the safety and efficacy profile. This can apply to interleukin 12 as well as a variety of potent immunomodulators like interleukin 18, interleukin 15 that can be linked using the FHAB platform. Interleukin 12 can orchestrate a robust immune response to many cancers and pathogens. Given the type of proteins and induced in the TME, such as the secreted protein enriched in cysteine called SPARC and GP60, several types of cancer, such as non-small cell lung cancer, melanoma, head and neck cancer, sarcoma, and some gynecological cancers are particularly relevant for this approach. SON1010 is designed to deliver interleukin 12 to local tumor tissue, turning cold tumor to hot by stimulating interferon gamma, which activates innate and adaptive immune cell responses and increases the production of PDL1 on tumor cells.
And finally, Pankaj, can you talk about the enhanced therapeutic effects seen in your preclinical studies and what you hope to show with the ongoing phase 1 program in adults with advanced solid tumors? And when can we expect that update?
Thank you for that, Janine. The murine form of interleukin 12 FHAB was shown to be much more efficient at blocking tumor growth compared to murine interleukin 12 wild type, while stimulating significant and prolonged interferon gamma production with minimal toxicity. Biodistribution studies in mice confirmed tumor delivery, and toxicological study in non-human primates allowed the submission of an IND and initiation of the clinical trials. As I said earlier, this has been demonstrated in a preclinical model where a 35 times better tumor shrinkage was observed as compared to wild type interleukin 12 at day 10 in a mouse B15F10 melanoma model. Recently, we have seen extended half-life, which is already published in human clinical trials and potentially showing that SON1010 targets tumor and is retained in the TME for a longer time frame. We expect to update on this phase 1 program for SON1010 in 2024 calendar year.
Thank you, Pankaj. And with that, this concludes the Virtual Investor What This Means segment with Sonnet BioTherapeutics. I'd like to thank Dr. Pankaj Mohan, CEO of Sonnet, for joining us today. And as a reminder, Sonnet BioTherapeutics trades on NASDAQ under the ticker SONN. And if you like what you saw today, I encourage you to visit sonnetbio.com for more information on the company, to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and events calendar. I want to wish everyone a great rest of your day.